Welcome to another episode of Progress, Potential, and Possibilities, discussions with fascinating people designing a better tomorrow for all of us. I'm your host, Ira Pastor.
Welcome, everybody, again to another episode of our show, bringing you not one, but two really fascinating guests today involved in creating a better tomorrow for many people out there. Today, we are going to be learning about ENA Respiratory, which is a clinical stage pharmaceutical company that is developing innate immune modulators for the prevention of complications associated with a range of respiratory viral infections in at-risk populations, including the elderly, also those that are at high risk of chronic lung infections with occupational risk, first responders, military, essential personnel. First, we have the honor of being joined by Dr. Ruth Tal-Singer, medicine development leader. She's an internationally recognized healthcare leader, clinical scientist with extensive R&D experience, including recent roles as Vice President of Respiratory Medical Innovation R&D, Value Evidence and Outcomes, and Senior Fellow at GSK. During her career, she led many projects involved in bringing new treatments to market. She also formerly served as the Chief Science Officer and CEO of the COPD Foundation, leading their patient-centered research, education, and global community outreach, including the creation of World Bronchiectasis Day. Also as leader of their digital health, medical devices, and therapeutics development accelerator network. She originally championed a relationship between the organization and ENA Respiratory. Dr. Tal-Singer has a couple hundred peer-reviewed publications to her name, did her PhD here in Philly at University of Pennsylvania in molecular biology and microbiology, moved on to do a postdoc at Worcester, and still lectures in the Institute of Translational Medicine and Therapeutics at Penn. We also have the honor of being joined by Dr. Scott White, Chief Medical Officer. He is an infectious disease physician. He spent the last couple decades working in the pharma and biotech industry, most recently as VP of Clinical Development for the infectious disease therapeutic area of Inovio Pharmaceuticals. Prior to that, was a physician project leader at GSK, leading numerous development projects there. And during the time at GSK, in collaboration with AstraZeneca, the EU Initiative for Innovative Medicines, was integrally responsible for establishing COMBAT, the European Antibacterial Clinical Trial Network, supporting the development of antibiotics for targeting drug-resistant organisms. Scott has his medical degree from University of Pittsburgh, did infectious disease fellowship at Penn State, and have that in reverse. And we're honored to have both of them. It's really great having both of you, and welcome to our show today.
Thank you.
Thank you. It's a pleasure to be here.
Yeah, it's you both have fascinating backgrounds, and I'd love to start things off just by talking a little bit about those backgrounds as we segue into what you're doing now. Ruth, I'd love to start with you. I mentioned a little bit of your background. I noticed that originally you started out studying herpes simplex virus and everything involved in that virus's entry to the human body. You continue that work at Worcester, and then when you got to GSK in the molecular biology group, you were involved in all sort of infection, inflammation, and immunomodulation, which are obviously three very hot topics today that we'll be getting more into. Take us a little bit into the background, if you would, and a little bit of the early days on your journey.
Sure, thank you. So I started my career actually as a critical care nurse, pediatrics, and I've seen a lot of patients. I've seen what the viruses can do to patients. At the time, it was herpes viruses. It was in the 80s when everybody was paying attention to herpes viruses. So I decided to pursue education and study the interaction between viruses and their hosts. So I think you see a running theme in my career, which is really understanding how we can protect people who are very vulnerable, be it children or people with underlying conditions, and protect them from infection. So that's pretty much has been the thread around my career.
Excellent. And Scott, you were a professor at Baylor, also at University of Pittsburgh School of Medicine. In addition to teaching, you arrive on also the scene at GSK, and you're into the antibacterial discovery unit, which, needless to say, and we've done a lot of episodes on this topic of drug-resistant organisms and the problems that are coming in the future and the fact that we have no antibiotics in development nowadays, or very few. Talk a little bit about those early days, because again, another extremely important topic as we get into this issue of prevention and keeping these bugs off and out of us. Obviously fits nicely into the story, but take us into your journey, if you would.
Sure. Yes. I mean, hats off to GSK for being one of the few large pharmaceutical companies that's still to this day trying to bring novel antibiotics to the market, despite the challenges of the commercial landscape for antibiotics, which it sounds like you've covered that to some extent, or touched upon it probably in early episodes. But that was something that I really championed and enjoyed during my time at GSK, and trying to bring novel classes of antibiotics to patients, as well as, and not just the science of the antibiotics, but also trying to work on the development landscape and trying to change how antibiotics are viewed by society, governments, and other institutions. And I'm happy to see that work continues, at least in some form, through the COMBACT clinical trial network that we've established over in Europe, and through several small companies that still kind of carry that torch for developing novel antibiotics. But I think I've taken advantage of opportunities now to move on to other areas where there's just as much need, and that's with ENA Respiratory, and trying to help those folks who don't have perhaps maybe great response to vaccines and need other ways to protect themselves against viral infections, and that's what got me excited about joining them a few years ago, and continues today.
Excellent, really excellent. And needless to say, respiratory viruses of all types, whether we're talking about the flu or COVID or everything in between, fall into that area of significant unmet medical needs. You are working again at this epicenter of all things inflammation, immunomodulation, in focusing on the innate immune response, and unfortunately, I guess a lot of what we heard, at least in the last couple of years, about the innate immune response, although we know it is sort of this first line of protection, a lot of what we heard the last couple of years is about hyperactivity of it. But needless to say, an extremely important barrier to dealing with infections. Take a little time just to talk a little bit about the innate immune response from the perspective of respiratory viruses, and a little bit about how the concept of INNA 051, what was the genesis of it?
So maybe I can go through, as a biologist, through that. So ENA 051 is a toll receptor agonist, and toll-like receptors are essentially pattern recognition receptors that recognize patterns of infectious agents that are not normal. One thing that is unique in terms of protecting the innate immunity, protecting us, is that the first barrier to respiratory viruses is the nose epithelium. And the concept around this intranasal approach is to prime the innate immune response, but not to activate it until it sees a virus. So a lot of the approaches that we've heard around the innate immune response are interferons or molecules that activate the immune response, which is our defense, our host defense against infection. The nice thing about this mechanism of action is that and we'll talk a little bit about the studies later, is that it primes the cells of the nose to be ready for any infection that comes in. But once the infection comes in, that is where those interferons and those antiviral responses are activated. So in terms of the molecular approach to targeting viral respiratory infections, is the concept behind this mechanism is to be ready. So we could use it as a seasonal prophylactic at times when viral respiratory infections are common, but we're not constantly activating. And as you said, we talk about hyper-activating the innate immunity. That is not the intention. The intention is to prime it. And when the virus comes, that is when the antiviral responses are activated, which is the data that we presented at a recent conference.
Scott, anything else to say on the innate front per?
Yeah, no, I think I mean that's the one beauty of how this product is intended to work, is that it's, again, agnostic to the virus that's coming in.
Sure.
It sends up the alert ahead of time, saying that as far as your innate immune system is concerned, and it does it in a way that some of the other toll-like receptor agonist or toll-like receptor systems don't, in that it doesn't immediately trigger on its own some of those responses that can make you feel uncomfortable, like the interferon responses. It just sets your body up to do that if it's needed, if it comes in contact with a virus, rather than preemptively launching all of those cytokines and having your body feel the negative aspects of those in the absence of a virus. So that's, I think that's the beauty of this particular toll-like receptor system, the toll-like receptor 2/6 system.
Excellent. Most recently, just a couple weeks ago, you announced data from a phase 2A study you did in influenza, and there was a couple of really interesting things here. One, it was a challenge study, and we actually local vaccine luminary Stan Plotkin, when he was on the show, had his talked all about challenge studies from the perspective of pan coronavirus vaccines. Very interesting to see challenge study come up here. Take us a little bit into I guess not just the study, but the unique nature of doing a challenge study. I know this, you organized about 123 adults in here, and then also the sort of the biomarker side of this, because in addition to obviously studying the prevention of influenza, looking at this from the immunomodulation perspective, you also studied a wide range of genes involved in immune response as a result of this study too. And it'd be interesting, again, to talk just a little bit about sort of the, I guess, the pharmacogenomics, let's say, behind this type of candidate.
Scott, do you want to talk about the clinical, and I can talk about the biology?
Yeah, let's talk about it. I mean, and these human viral challenge studies, they're a very useful tool. They can be a very useful tool for early development for a variety of interventions, whether they're vaccines, small molecules, or in this case, an immunomodulator. But they're also they're not always an exact science, in that you're still dealing with people who have been vaccinated, who have been exposed to, in this case, respiratory viruses throughout their life. And you look for people during the screening process for those who should be susceptible to infection with the challenge strain of virus, but are otherwise healthy enough that they know they won't get extremely ill from it and be a danger to them. But even that assessment isn't perfect. So there's some variability that's accepted with the model as far as how many people you bring in and then challenge with that virus, but actually how many of them might get sick or may not get as sick based on how much pre-existing immunity they might have that could interact or assist in preventing the infection with that challenge strain. So, and that's something that we encountered with our study, is that even though we do, the best job was done as possible to screen for people who should be susceptible and get symptomatic with infection from the challenge strain. In the end, the proportion of people in our particular study who got infected, as you could detect by nucleic acid methods looking for virus replication, as well as those who got symptomatic from that infection, was a lot lower than might be typical in other rounds of using this challenge strain in this type of population. So what that meant was there weren't quite as many events as we had anticipated that our drug would then have the opportunity to impact. Mm-hmm. But even without that, we still had evidence within the study of an effect of 051 at the higher dose on reducing that duration of virus production and a trend towards improvement of the symptoms. But I think what was really important, and Ruth can touch upon this maybe in a bit greater detail, is it allowed us an opportunity to really get down to the mechanism of action and the way these toll-like receptors work in triggering gene responses mm-hmm. in advance of infection and setting things up rather than waiting for infection naturally happening to set up those gene responses. And that's something that we were able to show in the study as well. So Ruth, if you want to go off on that a little bit further.
Right. Yeah, so from a technical perspective, these are nasal swabs looking at the expression of 730 immune genes that were studied. And what was observed is that there was, as I said, the concept is triggering or priming the immune response. There was an early increase in the expression of genes that are involved in the influenza host response. And there are known genes and known proteins that have direct antiviral effects against influenza. This was an influenza challenge study. So the early triggering of this gene expression, especially interferon genes, which is a good example, was consistent with the accelerated clearance that was observed in the clinical component of the assessment, and also linked the data very nicely to the phase 1 studies that showed the priming of the immune response. So it's the same genes that get primed. And then once the influenza came in the infected individuals, we saw the antiviral gene response. So it really ties nicely the human data with the preclinical data in human tissue culture, in cells of airway epithelial cells, and all this came together very, very nicely to explain the results.
Absolutely. And as far as, this was, I said, right, this was quite recent phase 2 study. Plans for sort of, specifically as pertains to, I guess, influenza moving forward. I noticed that obviously INNA 051 is building standalone, but I also see stuff in the materials about how potentially has sort of this vaccine adjuvant effect as well. If you could just take us through a little bit about sort of what's next specifically on the influenza front with the company.
So maybe I can say what's next. So one thing that we communicated recently is the investment by the U.S. Department of Defense and right. also the support that ENA Respiratory has from the Blue Knight initiative, because the whole concept is to have something that's not specific to one pathogen. And all the studies that were done up till now were with a liquid formulation. The investment that ENA Respiratory received from the Department of Defense is to enable the development of a more stable dry powder formulation. Okay. So the next study is the phase 1 to test the safety, tolerability, and pharmacology of the dry powder formulation. And really, the biggest question is now we have to take it to community infections. So Scott said, influenza challenge, and as you heard in the previous podcast, the conversation, this is a challenge experimental medicine study. Now we have to take it to the community and say, in a seasonal prophylaxis, what protection do we get? So that's really the plan in terms of next steps.
Excellent. Yeah, I noticed the BARDA relationship and Blue Knight and we had Sandeep Patel on several months ago from the BARDA Ventures group, talking about but where we need to be in terms of pandemic prevention, and it seems like clearly obviously we're putting a lot of effort behind vaccines, and that's great, but some of these other approaches obviously are synergistic, let's say, to preventing, God forbid, the next nasty thing from
we don't know
jumping.
what it's going to be.
Right.
Hopefully nothing, but what we've learned from COVID-19 is the impact that viral respiratory infections have. Not that they didn't have that impact before, but certainly the public is more aware now of what a viral respiratory infection can do.
Mm-hmm. And just wondering, as because we're in an odd era now where there's so much sort of vaccine hesitancy out there. I mean, do you get the maybe feedback? I don't know how early in the process, the market research and stuff like that going on, but that interventions like INA 051 may make people a little more confident, let's say, that have been so vaccine hesitant the last couple of years into believing in biotech approaches for these types of bugs.
Maybe I can comment on the survey that we did while I was still at the COPD Foundation.
Sure.
We basically asked patients, it was a survey in about 360 patients with respiratory disease, either COPD, asthma or bronchiectasis, and NTM lung disease, really asking them, first of all, we asked them whether they're vaccinated, and these are very compliant individuals. They know what viruses can do to them. So about 90% of them are vaccinated against influenza. That was what was available at the time in COVID. But what they said was they expressed, about 80% of them expressed very, very strong interest in having this intranasal prophylactic to protect themselves, because a lot of them, and even 1 in 8 in the survey, said that they ended up in the intensive care unit after viral respiratory infection. So within a population of people that are susceptible, there may be less hesitancy to take something that could protect them from what to them is a misery. It isolates them. They can't see their grandchildren, or they can't go to work. And that is a dramatic impact that viral respiratory infections have. So I think you're right that maybe there's a little bit of vaccine fatigue as well, because there are a lot of vaccines coming out, and patients are trying to decide what to take first. But I think there may be more awareness and more interest in protecting ourselves from viral respiratory infections. Mm-hmm. Scott, would you agree with that?
Yeah, absolutely. And I think, the fact that this is a nasal spray that you can be used at home is, the eventual intent. It's not something that's got to be administered in a pharmacy. It's something that people can do in a much more, in a simpler and more familiar manner. And it's something that can, it doesn't replace other interventions, but it is certainly complementary to anything else that these folks are doing. Vaccines, if they need to take other medications, even intranasal steroids, things like that for allergies, right, and other things. All of those kind of things appear compatible for use with ENA 051 nasal spray. So I based on this survey, and I think I think the profile is something that I think people could be fairly comfortable with. And I think along with it also, I mean, thus far in our first in human study, and then again in this influenza challenge study, the safety and tolerability so far has been pretty, I'll call unremarkable. Some people get a little sense of nasal congestion, maybe a bit of a runny nose, but when you do anything objective to look at flow of air through the nose, it's no different than those who got placebo spray. Mm-hmm. So I think it's continuing, hopefully, as we expect to see through the development, that fairly clean safety profile will just, I think, further reaffirm the potential for this to become something people could be comfortable with using relative to vaccines, where I know there's some hesitancy that still lingers out there.
Yeah, I neglected to mention in the intro to the show that I believe ENA Respiratory is headquartered in Australia. Both of you are here on the East Coast near me, I think. What's the? Can you just say a couple words about the company in general? I mean, obviously this is a huge market here in the U.S. Can you talk just a little bit about what ENA looks like, ENA Respiratory looks like from a global perspective right now, sort of where all the activities happening and what happens where?
Sure. So ENA Respiratory is an Australian company. It's based in Melbourne and Sydney, and that's where the permanent employees are, and the preclinical work is conducted in collaboration with a lot of advisors outside of Australia as well as within Australia. The clinical team is in the United States. The funding that's coming in from Department of Defense is US and BARDA. So the clinical team, Scott, as well as Bruce Miller and Courtney Krim, were all in the United States. And we recently partnered with a company in Ireland that's helping us with innovative trial design. So it's pretty much kind of an international effort towards the same goal, but the company itself is based in Australia.
Yeah, I would say, one of the things that, if anything, was positive coming out of the pandemic is that ability for people to work remotely or virtually. But I think it's opened up opportunities for companies like ENA to be able to bring in people globally who are experts in the field to help them bring these, this product forward, this technology forward. So, as I say, and I think, and we continue to leverage that opportunity any chance we can for trying to find the best people out there to help us bring this forward.
Yeah, and there was a very nice article recently on sort of highlighting it was actually, I think, Forbes Australia, talking about sort of the Australian US connection there. So no, it's obviously a very exciting program, clearly sort of a novel approach to what we have going on currently. And it's going to be interesting to continue to follow the company, and as you continue with these trials, not just influenza, but in a range of these respiratory disorders. Is there anything else coming up that we should know about per 2023 as we get into 2024 in terms of scientific conferences, presentations, places that either of you are going to be, where we can run into you, meet you, listen to you talk about the candidate or other research you're working on? Please, anything else I missed?
So the CEO, Christophe de Maison, and I will be at the ID Week in Boston in October. That's the next conference that we're going to be at. But normally, I think we will have a presentation at the American Thoracic Society in 2024. And that's pretty much in terms of conferences that I'm aware of that we will be attending.
Got it. Well, it's an exciting story. I will continue to follow it as you make this amazing progress that you've been making. For everybody that is going to be listening to this particular episode of our show across the podcast networks or watching on the YouTube channel, you've been listening to Dr. Ruth Tal-Singer, Medicine Development Leader, and Dr. Scott White, Chief Medical Officer, ENA Respiratory, doing really amazing things to combat a range of respiratory viral infections. Scott, Ruth, I want to thank you for both taking time out of your schedule to come talk to us for a little while about what you're up to. Obviously, thank you for doing it. And as we like to say on our show here, thanks for creating a better tomorrow via the type of work you do. An extremely important area, and wishing you the best with all of it.
Thank you.
Thank you very much, Ira.